So it's kind of a quick overview, and I want to talk about immunology in the context of complementary and alternative medicines. And so I kind of want to look at whether it's beneficial to the immune system, whether it's a fraud, or whether it actually injures your immune system. So I'll talk a little bit about what complementary and alternative medicines are, then who uses them, how many people use them, why am I studying this, and then what part of that do I actually study. Talk a bit about the immune system, do a quick overview as fast as I can, and then get into a little bit of our data. And then I want to spend some time with giving you a take-home, personally, of what you might want to think about for this. And then I also try to think of some things that you might be able to take back to the classroom. So that's kind of where we're headed. So what is complementary and alternative medicine? The National Institutes of Health defines it as a group of diverse medical and healthcare systems, practices, and products that are not presently considered to be part of conventional medicine. So we've all probably heard about these things. We might have used them, things like acupuncture, even though that's being taught in medical schools now, Tai Chi, yoga, herbs, things of that nature. And so I wanted to put in some poll everywhere, so I thought you might be able to do this on your cell phones, but I'm not sure if you'll be able to now. If you can go to this website, then you can click on here, and my question is, what percent of adults in the United States actually use or claim to use complementary and alternative medicine? So anything outside of the norm. So it's pollev, POLLEV.com/Christopher T132. pollev.com/Christopher T132. And the question, what percent of U.S. adults claim to have used complementary and alternative medicines in the past year? If I were doing in the same class, I'd hide your answers so you don't all influence each other. It's blank. Next one should work better. Sorry about that. So I kind of have this broken down into the top 10%, the next 15% or so, all the way down to 50% or below. And we're getting a pretty wide spectrum, lots of guesses. No one thinks it's 90 to 100%, it looks like, which is pretty reasonable, but the guesses are spread throughout. It's actually about 62%, according to a study. Now, the study comes from 2004, so the information is a little bit older, but it's the most comprehensive study I've seen. It was interesting, too. Women tend to use this more than men. The more higher educated you are, the more likely you are to use it, which seems counterintuitive sometimes. The sicker you are, the more you're likely to use it. And the more pain you're in. Pain is a great motivator for using anything outside of normal medicine. I have a family member with Crohn's disease, and we've been joking lately as he starts seeing an acupuncturist that I think he'd spread vegan mayonnaise on his forehead if he thought it would help the pain. So people who are in a lot of pain are ready to do anything that it might take. So 62%, almost 2/3 of adults in America use this. This is a multi-billion-dollar business. So what do you think the most commonly used forms of complementary and alternative medicines are? This one's a bit easier. You can text Christopher T132 to this number, 22333, once to join, and then you can text your message. So I mentioned a few already: Tai Chi, acupuncture, herbal medicines. This is great. When I use this in class, sometimes I'll get responses like, no. So this is great that all the words actually relate. That's wonderful. Good, so we're kind of thinking acupuncture, maybe yoga seems to be the most likely to be used. Certainly some herbal remedies, vitamins I saw here, herbal vitamins. some aromatherapy, but we think acupuncture and yoga are the most used. Actually, no. So these are the top 10. So I'd like you to arrange these now that I've given you the answer. So we were on the right track. We had some that were right. I'd like you to arrange these and rank them from top to bottom of what you think is the most commonly used. So there are some good guesses. We didn't quite hit all the top 10. We completely forgot about prayer. Is this one not working on the... It works okay.
I don't know what to do. I was dragging my whole phone.
So it's kind of interesting when you look at the top 10 too. We have prayer for yourself, prayer for others, being part of a prayer group. You have herbal remedies, which is one we hit. Yoga, which is one that we hit. We missed things like meditation and deep breathing. And diet-based therapies like going on paleo diets, becoming gluten-free, things of that nature. We missed chiropractic medicine. So we think that the most used are natural products like herbal remedies. We think yoga is number 2, paleo and other diet-based therapies number 3, all the way down to participation in a prayer group. We apparently don't like prayer as a group here, so that's okay. We're in a Jesuit school, but you don't have to rebel. So really interesting, and again, this is a study that's 13 years old, right? But a lot of this still holds true. You're way off. You're a very selfish group. About half of the participants said that my complementary and alternative medicine is when I pray for myself. Crazy. And then a quarter of the population is like, you know what, I'll pray for you too. Natasha's had a rough day, I'll give her a quick prayer. And these diet-based therapies that we thought would be much higher, only 4% have responded yes.
Just a quick question. What made up the basis for this group?
Oh yeah, age and location, because I think that makes
So it was adults nationwide, over the age of 18. I don't remember exactly. The sample size is pretty large. Yeah. So, and again, this is a 13-year-old study, so acupuncture may have gone up, yoga may have gone up, I don't know, but this is still pretty impressive. And it includes some things that we don't think about much. So, though this seems a bit crazy, there has been quite a bit of recent interest by the government, the National Institutes of Health specifically. So they formed the National Center for Complementary and Integrative Health, which used to be the National Center for Complementary and Alternative Medicine. So these are their 2 logos that they used to have. And the NIH had a really a lofty goal. They want to create functional integrative medicine. So if you're having back pain, can I give you acupuncture and a steroid and see better results? Right? That's what their goal is. But it's really hard to get to that goal. The treatments are not regulated by the FDA. So anyone can sell you an herb of anything with no proof that it does anything. And as recent studies have shown, we don't even know that what they say is in the pill is actually in the pill. It's a lot of cellulose, sawdust, things like that. It's crazy what they found in here. There's a very high consumption. Again, about 2/3 of adults use some form of this in the United States. That makes it a bit harder to regulate. There's relatively low funding for studying this, and so if you don't have good funding, you don't have good science, you can't make wise decisions. Now, the funding and research is beginning to change, but very, very slowly. So why am I studying this? Well, I'd like to answer that question by first giving a very brief overview of what I studied as a graduate student. So I worked at the University of Iowa, and I drew blood from human patients, and I used that to get macrophages out, which are a type of white blood cell, and I looked at tuberculosis, or mycobacterium tuberculosis, which causes the disease tuberculosis, and then I studied the lipids in that by using radioactivity, and then I wrote papers that looked like I took a game of Boggle and just threw it on the table and wrote those letters down. None of this is conducive to undergraduate research. They are not touching a patient with a needle. They're not touching an incredibly infectious disease. They're not touching radiation. and the word sphingosine kinase would make anyone avoid my lab, right? So I needed to change something. And I knew that coming into when I decided I wanted to do or have a career in higher education at a small university. So I knew I had to shift my research at Loyola. I knew I needed to involve the innate immune system because that's what I learned about. That's what my expertise is in. I wanted to involve undergraduates. I think it's really important for them, and they're my only cheap labor while I'm here too. I needed something relatively safe. Whenever you're working with cells and bacteria, there's some element of danger, but I'm not going to have them work with tuberculosis. I'm not going to have them work with radioactivity. I also need something that's publishable. So it has to be interesting enough that I can publish, but not so interesting that the people who can move faster at Johns Hopkins or Maryland or places like that can scoop me. What they can do in a couple of weeks, it's going to take me years to get done. So I decided complementary and alternative medicines might be a great place to do that, though. I think it was the intersection of all of those things. So I took a look at this list, and even though I'm at a Jesuit institution, I didn't know how to pray over my cells with a good control. So I decided to focus on natural products. We're still talking 1 in 5 people reported that they use these. And so I looked at echinacea and green tea, and now black tea, and the innate immune system. Keep talking about the innate immune system, so I'm going to give the fastest tutorial of that I can. I'm a horrible artist. There's a reason I'm in science. I'm not great at PowerPoint, so this is terrible, but maybe memorable because it's so terrible. So you're cutting meat, and then you slice your hand. That's horrible, because you've just got dirty meat germs all over your hand. So here's our blood supply, here's our layers of skin, and here's our terrible bacteria. The cells that were just cut open release a signal saying we have a problem, we have damage, and we have an infection. And they release small chemical messengers into the bloodstream. These distress signals propagate, move into the bloodstream, where they first interact with cells called neutrophils, which are awesome. Like the Incredible Hulk, they turn turn green when they get angry, and they actually make bleach. How cool is that? You have cells that squirt bleach on infectious agents in your body. It's amazing. So these guys freak out a little bit, and they get activated, and they run off to fight this infection. If they get the infection, you'll notice the bacteria are propagating already. They leave the bloodstream and they get into the site of infection or injury. When they get there, their signals combined with the signals from these injured cells and from the bacteria themselves induce inflammation, heat, pain, redness, and swelling. We've all experienced this when you sprain an ankle, when you have a zit, anything like that. This is just a larger inflammation that I'm representing here. So the neutrophils are there, they're starting to kill the bacteria. Macrophages come in next. This is the cell type I've spent the most time working with. So they come in next. They're eating. See what I did there? Pretty proud of myself. So both the macrophages and the neutrophils are phagocytes. Phago means to eat, cyte means cell. So these are eater cells. They're trying to gobble up these bacteria and kill them. While that's happening, platelets also come in and we plug volume. So we have our macrophages there. Neutrophils have left or killed themselves. Now dendritic cells move in. And if we zoom in on what these guys are doing, again, they're eating the bacteria, destroying them. But the macrophages and the dendritic cells do something else that's really important. They take that eaten bacteria and they put a chunk of it on their cell surface and they show it off to the adaptive immune system. So that's where we get our B cell response, our T cell response. That's the basis of vaccination. I'm not interested in that part. I like this first part. So then the cells, once they've eaten the bacteria and they have their antigens on the surface, they leave. They go through either the lymphatic system or the blood. They go to what we call the draining lymph node. In this case, if you cut yourself on your hand, the draining lymph node would probably be around your elbow. So they go there, and there they teach the other cells what the problem is and how to fight it. Quick immune system overview. We good? Any questions?
That was very good.
So, our experiments then, again, are looking at green tea and echinacea. What do these things do, if anything? Right? There's tons of people out there saying that green tea is like the cure-all. Echinacea is sold in everything, from airborne to echinacea pills. So do these things actually do anything or not? And I really wanted to focus on the activity of macrophages because that was the cell that I was most confident in working with. So we'll start off with green tea. Green tea is part of the plant Camellia sinensis. It's a flowering plant native to South and Southeast Asia, and it can make all the different types of tea. So white, oolong, all of them come from the same plant. They're just fermented for different lengths of time. So white is the least fermented, green is next, and black teas are the most fermented or the most oxidized. They're marketed for their antioxidant properties, which we all think is a cure-all for aging, right? Oxidants are actually really important to killing bacteria. That's why I thought this was interesting. That bleach that I talked about our neutrophils making, that's actually an oxidant. So do antioxidants prevent that from happening? That was part of the thought process. There are, of course, differences in brewing these teas as well, how long you steep them, what temperature, etc. So the research to date has really talked about EGCG. It's a catechin that appears to be the primary bioactive component. We don't think anything else in the green tea is having an effect. There are some other polyphenols that fall into this category, but this really seems to be the primary one. We know it does a lot of great things. It protects against certain types of cancers, slows the growth of other types of cancers. We know that it's important in macrophage function. So these are some pro-inflammatory things. NO here stands for nitric oxide, so that's another oxidant that's going to help kill. We know that it can help kill certain bacteria that can cause pneumonia and other terrible diseases. And there was one really cool study that showed that green tea, and they didn't even use fancy, they used Lipton green tea, destroys one of the primary components of anthrax. The Department of Defense was super excited. Now, so we're in the desert, you throw anthrax at me, I brew a cup of tea and I'm fine. So there's a lot of interest in this, but not a lot of basic science has been done for those reasons I mentioned earlier. Echinacea you've seen growing by the roadside without knowing it probably. It's purple coneflower. There are many different varieties. I focus on Echinacea purpurea. It's a perennial prairie wildflower, also known as purple coneflower. And the products of Echinacea, whether it's from the roots, the leaves, anything like that, is one of the largest contributors to the multi-billion-dollar industry that is complementary and alternative medicine. The unregulated multi-billion-dollar industry. So Echinacea has been, again, touted to be a bit of a cure-all. It's been suggested that it helps with treatment and prevention of cold and flu, that it can help with wound healing, can help with skin infections. But the results in all these cases are conflicting. And there's lots of reasons why. Some are that there are different preparations, that there are different parts of the plant that can be used, etc. In general, there is a consensus, though, that the alkylamides are the bioactive components. When we look at cold treatment prevention, there's a fair consensus that it does nothing. And for immunomodulation, there's a mixed report that it does both pro-inflammatory, so enhancing the immune response, and anti-inflammatory, or dampening the immune response. And again, these come from differences in study design. So we decided to approach this a little bit differently. Most people do an ethanol extract or an alcohol extract and put that on cells. We don't know many people who inhale, snort, or inject echinacea, so we decided to use a simulated digestion protocol. So we made some simulated gastric fluid, which is some salts, some acid, and some digestive enzymes. Mix it up in that for a little bit. As you pass out of the stomach and go into the intestine, we neutralize it with the help of the gallbladder, right? So here we neutralize with sodium hydroxide. It's just a little bit easier, faster way. We have some simulated intestinal fluid, again, some salts and some enzymes. We centrifuge that out to get all the chunks out, thinking that the chunks wouldn't pass the intestinal barrier, they'd be eliminated, and we took whatever was left, we sterilized that, and we added that to our cells. We think this is a more physiologically relevant way of approaching the question. And then we treated the cells with the equivalent of 1, 2, or 6 pills a day, which is what the bottle called for. That in and of itself is crazy. Unproven medication or herbal remedy, and they're calling for 6 pills a day. So I want to start off with green tea. And so SD is our simulated digestive fluid alone. There's a lot of junk in there. There's enzymes from pig pancreas. There's a lot of different things that could stimulate our macrophages. So this is just 30 minutes with the simulated digestive fluid. We've added some bacteria, and we're going to use this as our control. Within 30 minutes of the addition of the simulated digestive green tea, we see a huge reduction in the amount of bacteria. Green tea really helps these macrophages kill these bacteria faster. By about 60 minutes, though, they've caught up to each other. So it helps it happen faster, but it doesn't seem to help it in the long run. Everyone see that? So phagocytosis is the process that we talked about where the macrophage sees that bacteria, grabs it, pulls it in, and destroys it. So that pulling in process, I thought, well, maybe that's happening faster. Maybe that's why we're seeing faster killing. Not really. There's a slight decrease, if anything, in the speed at which the bacteria are pulled into the cells. So that wasn't the reason why. So then I looked at cytokine secretion. Cytokines are small chemical messengers, things like TNF-alpha, IL-6, IL-10. You may have heard of some of those. These are interleukins. Leuko referring to white blood cells, so these are things that communicate between white blood cells. TNF stands for tumor necrosis factor, so we first found it, noting that it would destroy cancer cells. Now, in green we have our green tea, and in blue we have our simulated digest. And what you'll notice in both of these cases, both TNF-alpha and IL-6 are pro-inflammatory. They're things that enhance the inflammatory response or the immune response. We see a peak earlier. I think that's what's causing this early enhanced response. But then afterwards they kind of level out, or in some cases, SD fluid alone beats it. But we get an earlier response. That could be important in patients that have a huge burden of bacteria. There were others that we tested as well. They didn't show much benefit. No change. So to summarize the green tea, we did see the increase in killing, and I should mention we were using Staphylococcus aureus. This is the organism that causes staph infections. It's one of the types that causes MRSA, or a variant of this causes MRSA. It's a really bad pathogen. Other bacteria that we used as controls were so easy to kill, the macrophages didn't have much of a benefit from green tea. If you're already killing 98%, green tea isn't going to help you do it any better, right? So this is the one where we really focused on because it's such a powerful pathogen, and we saw an increase in killing about 40%, but only at those early time points. No major impact on phagocytosis. If anything, phagocytosis was a bit slower. That pulling in of those bacteria were a bit slower. We saw a slightly faster release of the pro-inflammatory cytokines, and we think that might be what allowed it to kill early, but since that wasn't sustained, the killing also wasn't sustained. Any questions about the green tea? So then for echinacea, again we did that same ability, the assay where we looked at the ability to kill, and again we saw a pretty significant decrease, about a 50% decrease in the number of bacteria. So it also enhanced killing. We thought maybe this is due to pulling it in faster. And there was a slight increase in its phagocytosis, but not much. So I don't think that this is enough to cause that drastic increase in killing. When we look at cytokines again, now we're looking at two different cytokines. This is our friend TNF-alpha, again, one that enhances the immune response. Huge increase by echinacea. Huge increase. And without echinacea, so this is our simulated digestive fluid alone, there's really no increase. What worries me is that when we looked at this IL-10, which is an anti-inflammatory, one that's supposed to dampen the immune response, because we need to shut off the immune response, right? Once you've cleared the infection, you don't want all those dangerous chemicals around. That's what this does. But it is also drastically increased. So your immune system is receiving very mixed messages. Increase the immune response, but decrease the immune response. A recent paper came out suggesting that everything that you see that echinacea does is due to the bacteria found in the leaves and roots. I tried to control for that by eliminating some of the components, lipopolysaccharide, from gram-negative bacteria, and that didn't seem to dampen this response, but it's still something to look into. So to quickly summarize echinacea, it does increase the killing, and again, this is with Staph aureus, or staph infection causing agent. It increases phagocytosis, but not a lot, depending on the study that we did, between 20 and 60%. We get these increases in pro-inflammatory ones, but we also get increases in anti-inflammatory cytokines. And some research I didn't show you, we've identified the cannabinoid receptor 2 as one that partially contributes to this, but it's not the sole receptor for this. Cannabinoid receptor 1 is the one that responds to marijuana. Cannabinoid receptor 2 is similar in structure, but does not respond to that, only responds to alkylamides from things like echinacea. So a quick overall summary. Both green tea and echinacea alter macrophage function. Both do so by increasing microbicidal activity and increasing cytokine secretion. Both alter phagocytic activity, though not by much. We get a slight increase with echinacea and a slight decrease with green tea. But again, both drastically alter cytokine secretion. Some other research I didn't show you is that echinacea and fever. I thought big that a lot of times you'll take these drugs when you're sick. You have a fever. They're actually antagonistic. So if you have a fever, it makes echinacea no longer work. So any questions about anything that I've presented so far? So I tried to come up with some take-home points for you then. So does green tea help to fight off infections? What do you think? Initially?
Early.
So if you have an infection, do you drink green tea?
How do both of these compare to standard antibiotics or other medications?
So far less good. So antibiotics are much better at killing. Yes?
So when you see these medicines that say use the echinacea if you have a cold, actually they're useless. You need to be doing it preventative, not
Right.
time of. It's not preventative.
So colds and flus are viral. And I don't work with viruses in my lab, just bacteria.
Okay.
Bacteria, I should say. And so in the context of staph infections, then echinacea is, as we saw, it can help decrease the surviving amount of bacteria. So it does help it. But yeah, you're going to have to be preloaded on that.
Okay.
Yeah, great, yes?
So the one graph that you showed us that had the 30 minutes compared to the 60 minutes all in one place, is that you measured it at 30 minutes and then again after
An
a full
additional
hour?
Right.
So then I wonder what it would look like if you did it 30 minutes and then you took a second dose at 30 minutes. Would you still see that same peak?
So these macrophages have been in echinacea or green tea for 24 hours. So it's kind of like long-term taking it. These cells, they're cancer cells from mice, so they double about every 24 hours. So their entire life, they've been in this medicine. This is to simulate what someone might do if they're taking green tea or echinacea every day. We haven't asked the question about what happens if you take it immediately after. We've done concomitant, and it doesn't have much of an effect. It's not fast enough to get through the receptors, initiate the signaling, cause all the changes in the cell, and cause a readable effect within 30 minutes or 60 minutes. So this is really, the benefit would be from preloading, if you will. We've seen massive changes, which I also didn't show, in the cell structure, in actin polymerization or the cytoskeletal structure. But again, that all comes within 1 to 6 hours after consuming the green tea or the echinacea. So it wouldn't be fast enough for that 30-minute second dose.
Okay.
Great question.
Because when you take a pill, it's like, okay, take one, and then take your second dose the next and the third dose the next day. So if you
Right.
let it through repeated measures, I wonder what that...
Yeah, that's hard to do in cell culture because the cells are constantly dividing. And so you have to do more short-term assays. That'd be great for animal experimentation.
What's the recommendation on that 6-pill dispersant? Every 2 hours, 4 hours?
It depends on the company.
There it is.
Yeah,
So
because there's no reason to say that other than I can sell these people 6 times as much. That's amazing.
Absolutely.
A lot of them will say like 3 in the morning, 3 at night with meals, but again, there's no real evidence to back that up. So back to my question, should you drink green tea if you're sick? It's hard, right? Yeah, I guess. Yeah, maybe. It's kind of the way I answered the question. But you should absolutely drink it. I mean, I know it tastes like grass, but it prevents cancer. It helps with diabetes. It helps with weight loss. It helps with sugar tolerance. It helps with so many different things. Yeah, you should just drink it. And if you are drinking it, then you're preloaded and you're going to help kill some of those staph off really early. Yes?
Is black tea known for having some effects? Or?
So that has been studied much less. We actually just started working on that in the past year or so. It does cause changes in cytokine secretion, though not quite as robust as what we saw with the green tea. It's brewed at a different temperature and for a different length of time, and it's been fermented and the antioxidants have been removed. So we do get a completely different set of molecules that are going into the system. But yeah, we're just on the beginning of studying that. Again, black tea, it's quite beneficial in everything I've read. Not quite as beneficial as a green tea, but still drink it. It's cheap and could benefit you. So, echinacea. Yes, I'm sorry.
Back to green tea, a question. Are you saying, when you say drink it, are you saying brew it ourselves? Because I feel like when I go to the store and I look on the shelf, I see all these million different types of green tea. And that Lipton green tea looks like it might be soda a little bit. It's probably got some sugar in it.
It doesn't matter. It doesn't. Now, we haven't done the test of adding milk or lemon or sugar. There was one study that added milk to the tea and they saw no change. The idea being maybe that increased absorption, but they saw no change in any of their measurements. So whatever you find, brew it. Don't buy the pills. Interestingly, we found that if you buy the pills, which are much more expensive, they do not have the same robust response. You actually get a decreased benefit from consuming the pills. It's really that hot water extract somehow benefits you more. Yes?
But all your research has been done in vitro, so
Correct.
In culture, so you've got them like actually in some sort of media.
Correct.
Okay.
Yeah, so this is all in vitro, all with cells. A lot of people have suggested a move into animal studies. I'll remind you about Loyola, and I work with undergrads, and some people have been very successful with that. I'm good just trying to keep my cells not contaminated by my students. So, that is certainly somewhere someone should go. I don't think that's right in my lab. I'm also trying to chase down the molecular aspects, which are going to be much harder to do in animals than they are in the cell culture. Yes?
I couldn't catch the data, but you said about interleukin-6 and TNF-alpha both going up for green tea, right?
For echinacea. No, I'm sorry. For IL-6 and TNF-alpha work with it.
So are you thinking about going backwards through that pathway? What are some of those other genes?
Yeah, so we've actually, and I didn't show this either, but we've done some of the genetic analyses and found quite a few genes that are upregulated by echinacea. We haven't done the same studies with green tea yet. It's incredibly expensive and time consuming, so we just haven't done it yet. Great question. Good. Yes?
How was your study conducted again? What type of study was it?
So we have macrophages that are in a cultured dish, if you will, and they're from mouse. They are cancer cells, so they grow really easily, and then we treat them with the appropriate amount of either tea or echinacea that's gone through that simulated digest protocol.
I mean, as far as getting your study together, how did your study get? Did you randomly pick people, or?
So this is not using people, right? So this is all just cell culture based, and that helps normalize it too. I know when we were in grad school using macrophages for tuberculosis studies, we had to use someone under the age of 30, and they did not have taken aspirin, ibuprofen, or anything in the past 48 hours. It gets much more complicated with people. So again, with the resources I have and the time I have, I felt that cell culture was a better fit and allowed me to chase down those molecular mechanisms a bit better. Great question. So green tea, or I'm sorry, echinacea. Do you take it? I say absolutely not. Because it's increasing both the pro-inflammatory and anti-inflammatory, and I get confused enough, I don't need my immune system being confused too about what to do. Based on the data that I presented, it is good to take as a preventive, but if you're sick, don't take it. because that's going to make matters worse. But it might be good as a preventive. I still think it's a waste of your money. I think you're better off drinking green tea, which is super cheap, or black tea, or white tea. Any of them, they come from the same thing. So those would be my take-home points. Any questions about that?
Earlier you had talked about antioxidants, and the bleach substance that's produced is an oxidant. So are those, in the graphs that you had shown, is that what they are fighting against? Like they're counteractive to the...
Yeah, so no, interestingly, the molecules are stimulating a signal transduction pathway that is not actually scavenging those oxidants. They're in fact enhancing the production of oxidants.
Okay.
So when it seems to be when you're sick and when it hits a macrophage or something like that, it boosts oxidants. When you're healthy, it scavenges oxidants from your system, decreasing the aging process.
Interesting.
Is kind of the thought. Yeah. Neutrophils are awesome. I mean, really, you should read about them. They get angry and they make hydrogen peroxide, which is already really cool. And when they get angry, their myeloperoxidase, which converts, or which generates the hydrogen peroxide, it turns green. That's why your snot turns green when you have an illness. The neutrophils are there, they're angry, they're making hydrogen peroxide, and then that gets converted to bleach, and they're squirting bleach all over everything. That's also why they have a 2-day half-life. They're squirting bleach all over everything, including themselves. So it's really interesting to learn that. So I try to think about ways that you can take this back to the classroom too. Clearly you're not going to be working with Staph aureus, you're not going to be working with cell culture, but I still think there are some ways that you might be able to play around with this. I think asking your students, how do you know if this works? You know, I remember the original ads for Airborne, and it said, these are created by an elementary school teacher. It has to work. And I'm like, I'm married to an elementary school teacher. I don't trust him with that. So how do you, as become an informed citizen, and how do you look at this and say, do I waste my money on this? Or does it actually work? How do you dive into the literature or dive in and find a good resource? So some of the good resources I'd suggest to you are the NCCIH website. So this is the National Institutes of Health branch that focuses on complementary and alternative medicine. They have a lot of good information. If you can, getting into the research literature is great, but it's really hard, especially for high school students. I get intimidated by some of the papers I read. Another great one, a former research student of mine works at the International Food Information Council. They have a lot of very easily digestible, pun intended, easily digestible information for you to learn about what the science says about some of these things. And I think wrapping that into some of your exercise in the classroom might be feasible. And then I thought maybe there's a way to fold in some critical thinking or some reasonable skepticism. And I'd like to offer 2 different studies. I didn't have a chance to print them out, but I will send them to Natasha and she can put them up on the website for you. The first is about anthrax. It's the one I mentioned before. It's a study in which they used Lipton green tea and found that destroys anthrax lethal factor, one of the 3 parts that make up the anthrax toxin. So asking a student what they think about that and whether or not they should drink green tea and what's the likelihood of getting hit by anthrax, all these things might be a chance for them to really think about things, or even have some creative writing or some sort of critical thinking experiment. There's another paper that I want to send you all about, and it's Retroactive Intersessional prayer. So they did a study in 2001. They took 3,000 patients who had been in the hospital previously over the past decade with a bloodstream infection. They divided the names in half, so 1,500 to one group, 1,500 to another group. One group, that 1,500, they asked a prayer group to pray for them. The other half was not prayed for. When they analyzed the data, the group that was prayed for had a lower fever, had a lower hospital stay, and shorter or less mortality. There's some things wrong with this study. But it was published. And I think that's a really interesting thing for the students to think about. This is a published study. It was peer reviewed. Is retroactive prayer really a good thing? I mean, you all can pray for me anytime you want. But it offers the opportunity for students to be real critical thinkers about what's being fed to them in this information-rich society, but not necessarily good information society. So those were things I thought we might be able to take back from this, plus some cool stuff about green tea and echinacea. Any questions for right now? Yes?
What is the bioactive component in echinacea?
In echinacea, they think it's the alkylamides, and there are a couple of different types of alkylamides that may be involved. I don't remember the exact chemical names of them. I've shifted away from echinacea because I just don't see a lot of benefit to it. So, just quickly, some directions I'm heading with green tea, looking at phagocytosis versus killing and some of the differences we saw there, exploring the cytokines more, exploring the fever more, looking at antigen presentation, that next step, different preparations including black tea, white tea, oolong tea. For echinacea, I'm actually kind of done with it, as I just mentioned. I don't see a lot of value in it. I do see value in trying to dispel the myth of paying for it, but I don't know how much further I'll take that research. So if I do, I'll look at cytokines a little bit more in the interplay between fever and echinacea, and then some different preparations, as I said, leaves versus roots, etc. And then I do have some data where we stressed some echinacea plants and saw whether or not that caused a change in its ability to modulate the immune system. I haven't finished analyzing that data yet, but I think that could be interesting as well. So before I wrap up, I just wanted to thank Natasha and the CEE program. It's always great to interact with you all. This is the second time I've been the one here at Loyola, and I really, really enjoy it. I'd like to thank, of course, all of you for listening to me ramble on for a while. I'd like to thank the biology department here at Loyola, and then my research students who contributed to this. And a lot of them are older from the echinacea studies, but I was really impressed by the impact that we can have by doing research. And it starts with you all, and hopefully finishes up well beyond their interaction with me. So Christina DeFelice is now a PA in Denver, Colorado. Olivia Zurich is a Ph.D. in California. Becky Sterritt is a Ph.D. in Ohio. John Jacoby is a physician's assistant in New Jersey. Jordan Asadi is a doctor of osteopathy. Megan Myers is a Ph.D. at the International Food Information Council. Allison Myers is a Ph.D. candidate at Wake Forest. And then Jane Atkinson is a senior here at Loyola, but we have high hopes for her too. So again, these are the people who did the work that I showed today, but I wanted to add this to stress what an impact you all can have. And especially in a place like Baltimore City, where my wife is an elementary school teacher, education is the ticket. That's how things are going to improve. You all are fighting the fight. I see her come home crying, every week. I understand how difficult it is. We want to be a support, but you are fighting the right fight. By getting students into science, by getting them educated, you are writing their ticket out of the situation they're in. So thank you very much. I think that's all of my acknowledgments. So thank you very much.